Hello and welcome to video 31 for Mr. McGee's 9th grade biology class at Arizona School for the Arts. Today I'll be talking about what molecular evidence can tell us about how life evolved. Molecular evidence refers to the analysis of nucleic acids or other large molecules to determine the evolutionary history of living things. Although many different types of large molecules are used, we are going to try to focus on two kinds of molecular data: gene sequence data and protein sequence data. Gene sequence data involves looking at the arrangement of nucleotides in important genes that a group of organisms share. Remember that genes are codes for making proteins that use 4 nucleotides: A, T, C, and G. These nucleotides store information. By comparing the sequence of these nucleotides in genes of multiple organisms, we can construct a phylogeny or test the accuracy of a phylogeny that was made using other kinds of data. Basically, when we compare the genes of species that we think are closely related, we expect that the sequence of nucleotides from each species should be very similar. Likewise, if two species are not thought to be closely related, their gene sequences should be less similar. The reason this works is because changes to genetic information occur through rare random mutations. These mutations are hereditary, and they're passed down from one generation to the next. In the same way that a mutation can be shared by related members of a human family, when we see similar mutations between species, we know that those species share a common ancestor. This image shows a computer program that's used to analyze gene sequence data. Here the genes for 12 primate species are entered into the program, which color codes the A's, C's, T's, and G's. Scientists can then scan the gene sequences and count the number of differences between each species. This information is used to construct a phylogeny that reflects the simplest explanation from the data for each gene. Scientists can then test this phylogeny by analyzing multiple genes or by comparing this genetic data to other kinds of evidence. Protein sequence data follows the same process as gene sequence analysis, except that instead of nucleotides, scientists use the sequence of amino acids in important proteins. Remember that all proteins are composed of 20 amino acids. Scientists studying the protein sequence data assign each amino acid a different letter of the alphabet. The sequence of these letters can then be analyzed easily. The slide shows the sequence of amino acids for an important protein shared by mice and humans. Differences are colored either red or yellow, while similarities are colored green. As with gene sequence data, scientists then use this data to create phylogenies between living species. This phylogeny was created from data for a protein called cytochrome C. Does it confirm or contradict what you already know about the relatedness of these organisms? That's all we have time for today. Remember to complete the comprehension questions for this video. And as always, here's your bunny of the day.